Okay, good afternoon to everyone. So I'm Dr. Prathiba from the Genome Institute of Singapore. So it's one of the research entities under A-STAR, which is one of the government research institutions in Singapore. So my lab focuses a lot on plant genomes, and we've started studying plant genomes only fairly recently, like over the past 4 to 5 years. So our lab is headed by Prof. Ravindran, who's the senior group leader at GIS, and we study plant genomes with a focus on food, biodiversity, and human health. So of course, we use some of the latest sequencing technologies, particularly the PacBio Hi-Fi, to assemble the genome. So these are about 50 genomes that we're working on, but we have like about 100 Southeast Asian plant species that we've already sequenced, and we're currently annotating the genomes for those. So the goal is to use the genomic information of these plants to eventually understand the molecular basis for their use in traditional remedies, because a lot of them are medicinal plants, as well as to understand why, what are the nutritional traits that they carry. We also use the genomic information to learn more about the evolutionary significance, as well as environmental adaptation of these plants. As well as to identify like, novel genes and pathways that regulate certain agronomically important traits. So most of our work at GIS is done in collaboration with the Singh Health Duke NUS Institute of Biodiversity Medicine, which is BDMed in short. So BDMed is a fairly new institute which was established in 2021 by Professor Ravindran himself. And so BDMed aims to drive and accelerate biodiversity research through 3 key domains of interest. So we have the herbal biodiversity and medicine, where we are focused more on traditional and herbal plants, particularly those plants that are used in TCM. Then we have the food biodiversity and nutrition. That's where we've sequenced some of the fruiting crops, as well as agronomically important crops like the leafy greens, particularly in Singapore. And then we have the urban biodiversity and wellness as well. So right outside the BDMed lab, we've actually established a genomic garden, which is probably the first of its kind, probably the first in the world. So all the 100 Southeast Asian plants that we're currently sequencing are actually in this garden. So it was established and curated by BDMed. And the choice of plants here, they were selected not just for their genomic intrigue, but also based on their translational potential. For instance, like the traditional plants that are used in used for herbal remedies and for human health, as well as agronomically important plants, and also those that are known to have very important bioactive compounds that have been hypothesized to have nutraceutical value. So we also do a lot of clinical studies where we test the phytochemicals from these plants to see whether they have anti-cancer effects or any other beneficial effects for human health. So how different is it to assemble a plant genome compared to the animal genome? Plant genomes can be quite complex. The reason being is the genome size can be pretty large for most of the plants. So for instance, the genome of wheat is like 5 times larger than that of humans. And just a few weeks back, there was a publication about a fern-like plant where they found that its genome is about 50 times larger than that of the human. So it's about 160 GB. And so that kind of increases the complexity when it comes to assembling and annotating these genomes. And of course, a lot of plants have multiple sets of chromosomes, so they're not always diploid. Like wheat can, there are certain cultivars that are tetraploid and certain cultivars that are hexaploid. And repetitive sequences, that really makes the assembly difficult, because in plants, more than 50% of the genome has like our repetitive sequences or the retrotransposons. And since plants tend to outcross or outbreed, the level of heterozygosity is pretty high. So this poses a lot of challenges when we're trying to distinguish the alleles, especially when we want to do a phased genome assembly. But with the advances in sequencing technology, like the 3rd generation sequencing technologies, we are now able to overcome these complexities and able to assemble high quality genomes for plants. And of course, we combine it with some of the chromosome confirmation capture methods like Hi-C or scaffolding techniques. So that helps us to create more comprehensive genomic maps. And sometimes we also add in the short reads in order to polish the final genome to get a very high quality genome. So this pipeline was carefully tested and identified and kind of works very well for us for plant genomes. So you can see that at the core of this pipeline is the PacBio hi-fi long-read sequencing, which we're quite reliant on. And we combine this with some of these scaffolding techniques like the Hi-C and Omni-C in order to create a genomic map. So by combining these techniques, we've been able to overcome or navigate the complex and repetitive nature that plant genomes have, and thus allowing us to ensure that the genome quality is pretty high. So why do we need a high-quality genome? Because to make novel biological discoveries, we need a really high-quality genome. You don't want to miss out any of the genes, and you want to have lesser gaps in your genomes. So these are two of the notable genomes that have been assembled and published in our lab. So the first one is that of the durian. So I'm sure most of you are familiar with the durian fruit and the stinky odor that it comes with. So in addition to assembling the genome, we also found out the genes or the pathways that are responsible for that stinky smell that emanates from a ripening durian fruit. And we also assembled the genome of the national flower of Singapore, which is an orchid species called the Papilionthemis joachim. And here we identified a class of enzymes that's responsible for the production of vandopterosoids, which are known to have a potential in reversing skin aging. Yeah. So I'll just talk a bit about the durian sequencing or the genome assembly that we've done. So this assembly was done back in 2017 using the PacBio SMRT sequencing. So back then, I think it was the RS2 machine. So here we used about 50 micrograms of the genomic DNA. We're able to create a 20 kilobase pair SMRT Bell library. And the average read length of the long reads was about 6.2 kb. So using scaffolding techniques, we were able to improve like the scaffold N50 to 22 MB. And you can see that about 54.8% of the assembly is actually repeat regions. So it's pretty high, the number of repetitive sequences in this genome. So what did we learn by assembling this genome? So like I mentioned earlier, we first started with like a comparative genomic analysis where we compared the genome with other species in the same family, which is the Malvaceae family. So you have the cotton and the cacao, which also fall under this family. So through that, we actually found evidence for a lot of gene duplication events. So we found that there was whole genome duplication events in the durian lineage after speciation from cacao. And then we also complemented it with RNA-seq analysis, where we did like a transcriptome study across different plant organs. So we compared the fruit organs with the non-fruit organs. And here we found that most of the genes that are related to sulfur, which gives that pungent odor from the durian, as well as the ripening and flavor genes, were highly upregulated in the fruit compared to the non-fruit organs. So we also identified a particular gene called the MGL, which is the methionine gamma lyase, which uses the substrate methionine to produce these volatile sulfur compounds. And we found that this gene is highly interconnected with the ethylene biosynthetic gene, which is ACS. And ethylene is a phytohormone, which plays a significant role in the ripening process. So we found that these two genes were interconnected, meaning the ripening and the odor pathways were interconnected, which has evolutionary significance because the fruit dispersal for durians is quite reliant on these odor-enticed primates. So the odor that emanates during the ripening process is basically to entice these primates to come and disperse the seeds, disperse the fruits and the seeds. Yeah. So this genome assembly that we did initially, the reference genome, has a lot of gaps, and it was also not haplotype phased at that point because in order to get a better accuracy and to improve the overall contiguity, we actually merged the haplotypes and got a single sequence. So now we actually moved on to get a T2T, telomere to telomere genome for the durian, where we used PacBio, but we also combined it with some of the ONT reads, as well as the Hi-C scaffolding. And here we were able to actually get a full-length T2T genome. You can see that there are no more gaps now. So the idea is to eventually pick out maybe some of the important genes that we might have missed out in the earlier reference genome. So most of the repetitive regions tend to be near the telomeres or the centrosomes, and it's also known that a lot of epigenetic changes take place in these regions, which could be important when it comes to identifying important traits. So the other important fruit that we've assembled the genome for is the sapodilla, or the sapota, or the chiku, as it is known. So there are about 21 varieties of the sapota cultivated in Malaysia, and most of the Southeast Asian countries are the major exporters for the sapodilla fruit. And it also has a lot of medicinal properties, like the seeds and the flowers contain tannin and saponin, which are known to have a lot of medicinal and anti-cancer properties as well. And of course, it's of commercial value, and that's one of the reasons also why we've assembled the genome for it. So what are the other reasons we assemble the genome for? Because we want to eventually do a pan-genome, assemble the genomes across different chiku cultivars as well as species to identify novel genetic variants where we can use them to integrate them into like the cultivated or commercial varieties to make them more climate resilient and more disease resilient, as well as to analyze like epigenetic modifications, which are important for some of the traits. So this is just KEMA analysis showing like how heterozygous the genome is. So the two peaks here shows that it's pretty heterozygous. And we did it both using short reads and long reads. Typically, the KEMA analysis is done with short reads, but we found that even the long reads was pretty accurate, and it was able to give a predicted genome size which was within 20% of the Illumina's predicted range. So this is just the pipeline of the genome assembly we used, where we used PacBio Hi-Fi, and then we used Hi-Fism, which is like a PacBio haplotype tool to get your phased genome. This was then combined with the scaffolding techniques in order to arrange them into the different chromosomes. And then finally, we have a phased assembly. So you can see the BUSCO assessment here, which gives you an idea about the completeness of the genome. Of course, there are still some missing BUSCOs because it's not a T2T. We still have some gaps in there. But the KEMA analysis shows that a very high QV value of 59. And from the assembly statistics, we know that it's not a very large genome. It's about 1.29 GB, and we were able to get a scaffold N50 of about 110 MB. So usually we complement our genome assembly with transcriptome or metabolomics to try to understand certain pathways or mechanisms. So here we try to study the ripening process in chiku. So we did a transcriptome or RNA-seq analysis of the two different tissues in chiku, namely mainly the pulp and the skin. So chikus are climacteric fruits, meaning they ripen after you harvest. They're able to ripen even after you harvest them from the plants. So day 0 is when we harvested them, and then we have the day 1, 3, and 5. So I just have some preliminary data analysis where we show that most of the gene expression changes take place within the first 24 hours. So when we compare between day 3 and day 5, there's not much changes in the transcriptome. So you can see that so the day 3 and day 5, there was no significant pathways, but between day 0 and day 1, we see that most of the plant hormone signal transduction genes and plant pathogen genes, as well as the MAPK signaling, which has been shown to play a role in ripening process, were all enriched. Sorry. So the next thing we're moving into is to do pan-genomes. So we're also trying to do this for the durian. So there are a lot of wild species. In addition to the commercial cultivars that are consumed, there are also a lot of wild species which are more disease resilient. So the idea is to identify genetic variants or novel genes which can be eventually integrated into these commercial varieties to make them more resilient, because diseases in these cultivated farms is a large issue in durian cultivation, and the losses can be pretty high because the durian is sold for really high prices. So any loss in the farms can be a big economic loss to the farmers there. So it's really important for them to increase the genetic diversity and to make them more resilient to diseases. So through the pan genome, we aim to identify like SNPs or structural variants that are associated with some of these important phenotypic traits or even nutritional traits. Like if we want to increase the nutritional content of some of these fruits or leafy greens. Yeah, so the idea, so currently we are actually doing pan-genomes for durian as well as one of the leafy green vegetables that's commonly consumed in Singapore. So this is just the different pathways that we would eventually like to achieve by starting with the whole genome assembly. So in order to identify genes or for disease resistance, biotic, abiotic, or even to improve the nutritional content of plants, I think the first step is to have the genome for the plant, because most of the plants that we assembling the genome for, they're all de novo genome assemblies. So without this genome information, it's very difficult to actually achieve any of this or even to do CRISPR editing. So that's why the first step is where we are putting most of our resources into. So I'd like to thank the GIS and the BDMet, all the members in the GIS and BDMet team who are involved in these projects, and all the collaborators who are part of this. So you can see we're all eating durian here. So our prof likes to take us out for durian eating quite at least once a year. Yeah, so I think it's his love for durian which made him to assemble the durian genome first and then move on to other plants. Yeah. So if you'd like to reach out to us or for any collaborations, please feel free to reach out to Prof. Tay. Or to me, to Cedric, who's the assistant director of BDMet, to me, or Abner. A lot of credit goes to Abner because he is mainly involved in a lot of the genome assembly and annotation projects. So if you have a lot of technical questions about the genome assembly pipeline, you can always reach out to Abner. Yeah, thank you.
Thank you, Dr. Prathiba.